Okay, so as Dennis mentioned, I'm going to discuss ecological genomics in the frame of diagnosing coral health. And I'm going to be talking about new technology that has been explored and is currently in use in various fields of coral ecology and coral health and physiology. So as much as I love talking about my own research, this is going to be more of an overview kind of presentation. So I want you to leave here with an understanding of, or my goal is for you to leave here with an understanding of what's happening in the field, what kind of research has been done, what still needs to be done, and I will sprinkle in my research throughout the talk as well. Anyway, so let's get started. So the outline of my talk is I will briefly overview some of the key concepts here that we're interested in. I will give a brief introduction to corals, one slide, because I'm sure a lot of people here already know what corals are. I will discuss what global climate change is and what kind of impacts it has across the globe, and what are these impacts on coral reefs, and what can we do to try to quantify these impacts or measure them. The second half of the lecture will be on the use of molecular diagnostic technology in studying coral research, health, and physiology. And the three areas that I will focus on are increased ocean temperatures and coral bleaching, coral immunity and disease, and ocean acidification and coral calcification. So what exactly are corals? Coral reefs are the largest biological structure on Earth, and they are visible from outer space. So this is a satellite image, and these kind of white specks, white line is a coral reef. All of that makes up coral reef. And the diversity that's seen on reefs has given them the name the rainforests of the sea. So without the actual coral colonies making up this reef, there wouldn't be the diversity that you see there. The corals make the foundation for the reef and bring in that diversity. And coral colonies create the structure of the reef, the topography you see there, that also supports the diversity. And this is a coral colony, and you can see all these little bumps are individual polyps. So this is not the same species, but this is what one of the ways a coral polyp looks. You have the tentacles in the mouth. And hundreds to millions of these little polyps form together to create a big colony. And then the colonies form together to create an ecosystem. And what we're interested in understanding is these global impacts that are happening. How are they affecting individual polyps? And then in turn, how does that impact a colony? And from there, once we identify how colonies are responding, whether there's partial mortality or full mortality or recovery, how does that affect the ecosystem? So what is global climate change? Global change is defined as a transformation that occurs on a worldwide scale or exhibits cumulative effect to have a worldwide impact. And climate change refers to significant changes in climatic patterns, such as temperature, precipitation, or the frequency and intensity of storms or El Nino events that last for extended periods of time. And during this lecture, I am not interested in discussing or debating the causes of global climate change. I'm more interested in understanding the impacts it's going to have, or the predicted impacts or current impacts on corals and coral reefs. So there are many effects of climate change, and they can range from having impacts on geomorphology, which is anything having to do with landmass, such as erosion or loss of coastline due to sea level rise. In addition, you have changes in the atmosphere that can include the changes in chemistry of the atmosphere and warming of the atmosphere. And this is a graph, the top part of the graph here is the differences in global mean temperature from the early 1800s to 2000. And what's interesting is not only the increasing temperature, but the rate in what the temperatures are increasing at, and particularly in the last several decades. That's what's very important, is how quickly it's happening. In addition, you have impacts on the hydrosphere, and the hydrosphere is any of the water that is on Earth's surface, including ocean and freshwater. And being a coral biologist, I'm more interested in the ocean, where we're seeing increased ocean temperatures and the potential for ocean acidification, which is directly tied to changes in the atmospheric chemistry. Then there's also impacts on the cryosphere, and the cryosphere is any frozen water, so any water that's tied up in kind of frozen forms like glaciers and the polar ice caps, which we know are disappearing and or melting. So what are the impacts on coral reefs, or what do we have to worry about with coral reefs? Well, as I mentioned, we're seeing increasing seawater temperatures, and this leads directly to coral bleaching and has an impact on disease, among other things. And we're also seeing an increased incidence and severity of coral diseases. And we're interested, because of the changing atmospheric chemistry, in ocean acidification, which is also a changing in the ocean chemistry, which causes it to have a lower pH. And I will discuss the implications for that a little bit later on how that's going to impact coral. And then we can see increased incidences and intensity of storms, including freshwater input, sedimentation, and increased exposure to pollutants and nutrients. However, I'm only going to focus on temperature, disease, and ocean acidification for this lecture. So how can we measure or quantify these impacts on coral health? Well, doctors use advanced technology to identify molecular mechanisms that are behind human diseases, such as cancer, chromosome abnormalities, genetic disorders, and protein dysfunction, which are the sub-mechanisms that are involved in a lot of different diseases. And the technology is very well defined, and there's a lot of work and money that has gone into developing it. So can we use this technology to diagnose coral health, and what exactly is this technology? And I do want to point out that I'm more interested in the results of this technology, so it's not going to be a very technology-focused lecture. I'm not going to explain what the technology is. I want you to understand how we're using it, really. But just to give a brief introduction to what it is, molecular diagnostics is using tools to measure DNA, RNA, proteins, macromolecules, and metabolites to detect abnormalities, mutations, and or biochemical changes that are associated with abnormal or disease conditions. So as I mentioned, the three things for the rest of the lecture that I'm going to talk about are temperature, disease, and ocean acidification. And we're going to start with looking at increased temperatures and coral bleaching. And this is a field of bleached coral, and it may look awfully pretty, but it's not very healthy. The coral is likely going to die because you can see a little bit of the brown coloration in the tissues here, but all of this white part is where the coral has bleached, and you're seeing the skeleton through the bleached tissue. So what is coral bleaching? Bleaching is the loss of symbiotic algae that lives inside coral tissue. So this is single cellular zooxanthellae, or the symbiotic algae that lives in coral tissue. This is a coral polyp, and you can see the brown spots all along the edges here. These are all zooxanthellae that are inside the coral tissues. And what happens during bleaching is you can see here a diagram of a coral, a healthy coral, with all the brown or greenish algae in its tissue. And upon bleaching, the zooxanthellae either leaves or is expelled from the coral tissue. And this can happen in as little as 1 to 2 degrees Celsius change of ocean temperature, especially if it's sustained. And corals can recover from bleaching. It's when it's sustained bleaching that the mortality is more likely to occur, and it also makes them more susceptible to disease and or other stressors. So what do we know, and what has been done so far? And what are the mechanisms of coral bleaching? There are multiple theories regarding the mechanisms of bleaching, ranging from oxidative stress theory to apoptosis and autophagy, which are different forms of cell death. There's also protective protein production by the coral host. And then there's a hypothesis called the adaptive bleaching and lipid production by the zooxanthellae. Due to time constraints, I am only going to focus on oxidative theory, cell death theory, and protective protein production today. So the oxidative theory of bleaching is involved in the production of reactive oxygen species. At elevated temperatures, damage to the photosystem of algae, the symbionts, because they're photosynthetic organisms. The elevated temperature causes them to release reactive oxygen species, or ROSs, which are known as hydrogen peroxides or superoxide anions. The accumulation of these ROSs damages a wide range of cellular components, including membrane structure, proteins, and DNA. And the corals respond and try to mitigate this damage by producing antioxidants such as superoxide dismutase, catalase, and peroxidases. But ultimately, if the ROSs are continuously produced and the temperature doesn't change or something doesn't happen, then the antioxidant system is going to become overwhelmed and bleaching is likely to occur. And this is similar to the response of human cells to certain types of cytotoxicity and aging. That's what happens. Oxidative stress significantly influences cellular aging. So this is just one of the types of technologies and or tools that can be used to understand these mechanisms. This is a type of algae. It is not zooxanthellae. It is an algae called Volvox that is aquatic. And what's happening here is using a fluorescent dye that binds to hydrogen peroxide, which is one of the ROSs that I mentioned. And these images are called fluorescent micrograph images. And where you see the red, the red fluorescence is wherever there's hydrogen peroxide. So you can see in this Volvox, you have localized production of hydrogen peroxide due to ROS releases at those locations. Whereas on this image, it's a little hard to see, but you can see that the localized areas have spread out and the red has become more of a cloud around the entire alga. So it's experiencing an increase in not just localized production of ROSs, but a global production of ROSs, and is likely damaging the algae and causing it to break down. So I have had my own experience with investigating oxidative stress in coral systems. And my research has shown not necessarily that it's a mechanism of bleaching, but it's more of a general response to any kind of stress. And I've done this using a coral microarray. So I have hundreds of specific genes all arrayed on one platform, and I can look at their expression under different conditions in the coral host or the zooxanthellae. And what I'm seeing is that one of the first lines of stress upon any stressor, ranging from sedimentation to low salinity or freshwater exposure to xenobiotics and even disease, is I see oxidative stress turned on. So I'm not convinced that it's necessarily a mechanism of coral bleaching, but just a symptom of stress. And this was also shown by Barrett et al. In a recent publication. This is a diagram from their research, or from their publication, and it shows, imagine this being the coral, and this is zooxanthellae, so the coral host, and you have multiple stressors coming in that hit the host, which then responds. There's oxygen radicals produced, and then you have the antioxidant system turn on, and it tries to shut down here the production of the oxygen radicals. And if it is successful, then it stops there. If not, it goes on through other types of stress response systems, which vary by the initial stressor. So what about the cell death hypothesis, also known as the apoptosis and autophagy theory? This was, recent research was done by Dunn et al. In 2007, and they identified that apoptosis and autophagy was a mechanism of bleaching in the symbiotic sea anemone Aptasia pallida. And the study used several molecular diagnostic techniques from the field of human health and other molecular research to investigate this, including metabolite pathway inhibitors, protein binding to shut down the function of proteins, and gene knockout or gene silencing so that the genes could no longer be expressed, to investigate the mechanisms, the molecular mechanisms that they were looking at. And the results suggest that bleaching could be a modifying immune response that recognizes and removes dysfunctional symbionts. And what I mean by that is that the coral host recognizes that a symbiont is dysfunctional, whether it cannot photosynthesize anymore, or it's producing ROSs, or it's just no longer a good viable zooxanthellae. And what it will do in autophagy is it will engulf the zooxanthellae. This is a mitochondria, so this is just here for an example, but it's the same premise. Imagine this is an algae cell. So it would engulf the alga fully and then lyse the alga so that it was no longer viable. It was non-damaging anymore. And the mechanisms for apoptosis are a little bit different in that what the coral would do would be to program its own cell death. So whatever cell had the dysfunctional zooxanthellae in it would be programmed to die and then just slough off. So those are a mechanism of known bleaching. So another mechanism is known as the protective protein production, and this is fairly well studied in a lot of model systems, including fruit flies and lab rats and humans, and it has been around since my early study of coral. I isolated heat shock proteins my first year in grad school, so I'm familiar with this mechanism. And it is essentially that corals will induce a suite of proteins that are protective and try to stop coral bleaching. And some of these proteins are heat shock proteins, fluorescent pigments, mycosporin-like amino acids, and antioxidant enzymes like I mentioned previously. And this is a diagram from Baird et al. In '09 as well. And it shows kind of a diagram of what would happen during light or heat stress, in that you have the stress coming in, hitting the zooxanthellae and the coral host, and that either mycosporin-like amino acid or fluorescent proteins or fluorescent pigments, which act as sunscreens in the coral, would be induced to try to prevent this stress from happening. And if it does, then the coral is nice and happy. If not, if the mechanisms are not successful or the stress is sustained or another stressor is involved, then photobleaching and photoinhibition will be induced and coral bleaching will occur. And these are some examples of protective fluorescent pigments in coral tissue that are produced by coral. And this first image shows, I know it kind of looks like a blob, so I'm going to walk you through it. These are also fluorescent micrograph images taken with a microscope. And in this big image on the far left, this is a coral that has been exposed to extended periods of intense sunlight. So intense, it has the heat, it has the sun, and it's just been exposed to that a lot. These kind of lumpy features up here are the tentacles, and the green is called green fluorescent pigment, and that is a photoprotective mechanism used by corals. The red spots are the zooxanthellae, the symbiotic algae, and they've all moved down to the base of the coral polyp, so that the photoprotective mechanism or pigments are all at the top, protecting the coral and protecting the zooxanthellae photosystems from the harsh sun and heat. Image D right here shows the exact opposite. This is a coral that was kept in the shade, and you can see all the red zooxanthellae are in the tentacles up at the top, trying to make use of any surface area they can get to photosynthesize, whereas all the protective pigments have moved down to the base of the coral because they're no longer needed for photoprotection. And the image on the right just shows the range of photoprotective pigments that are available in coral, ranging from a pretty blue at the top to greens and yellows and reds. So it's a pretty neat mechanism that they have to be able to produce their own sunscreen and thermal protectors. So all of these examples center around the host response to bleaching. But what about the zooxanthellae? Preliminary research in my lab indicates a possible mechanism of zooxanthellae response to external stress as well as internal stress that the host produces, so that they're not just the ones causing the problem with photo damage, but they also are responding themselves to the situation. This is an experiment I did in 2005 and 2006, and I'm not going to get into the details of it. Really what I want you to see here is one of the results that I found that was a little bit surprising to me. So let me just explain what you're looking at here. This is a graph of averaged gene expression by the coral over dates a period of time. So each one of these periods represents a sample at a specific site and during a specific period of collection. So each one is a different coral. And the green line is the average of the zooxanthellae gene expression. The yellow line is the average of the host coral stress response. So just the stress response, so only the genes that are expensive for them to turn on, and they only turn them on under unusual circumstances because it's energetically costly to express them. The blue and gray lines are just normal cellular functioning genes that kind of turn on and off with the flow of normal cycle. And what's interesting to note here is you see two specific stress events in these samples and these samples where stress gene expression in the host spikes. And it's interesting to note that the zooxanthellae gene expression correlates at a 0.9 correlation analysis with only the host stress gene expression. Well, what were these genes that the zooxanthellae were expressing? 3 of the genes were involved in photosynthesis, and they decreased with increased host stress gene expression, indicating, again, an impairment in the photosystem. They were PS2D1 and a chlorophyll binding gene and a UVB inducible gene. And this might be thought to support the oxidative bleaching theory, but it doesn't because none of these corals were bleached. They were stressed. So it supports my hypothesis of a generalized stress response seen by oxidative stress or ROS production. Another gene that was turned on that's indicative of stress is rubisco. This catalyzes the first step in carbon dioxide fixation, and when it's highly upregulated, is usually an indication that plants are experiencing a level of stress, and it can be increased by multiple types of stressors. But the interesting one is the SMCAM1 gene, and this is a flagellar motility gene that increased in this experiment with host stress gene expression. And this is interesting because in a symbiotic relationship with coral, zooxanthellae lack flagella and are in a coccoid state. When they're free living and they're swimming about on their own in the ocean, they have flagella and are in a gymnodimnoid state, as you can see here. So it's kind of interesting, why are they producing a flagella gene when they don't have or need flagella because they're not moving around in the coral tissue? So this could be an indication that oxidative stress represents a cellular signal initiating a breakdown in the coral-algal symbiosis, and that the algal symbionts may be trying to actively leave a situation that's no longer beneficial for them, or that maybe the host environment is no longer good for them anymore. So they might be trying to get out. And this is preliminary research and requires some more study, but I'm very interested in pursuing it further. All right, so now I'm going to talk about coral disease and immunity, and what is known in regards to disease, and what have we been doing with understanding the mechanisms of coral immunity, especially when it comes to diseases. There are several theories regarding coral immunity and mechanisms of disease, and these include innate immunity, just kind of naturally there, and innate cell immunity. There's also a holobiont probiotic theory, and this is the ability of the holobiont to kind of produce antibiotic compounds to keep disease bacteria at bay or away. And then there's another theory called the symbiosis suppression of coral immunity, which will be discussed last. And before I move on, I will discuss a little bit about my research here studying coral disease, and I'll talk about Vibrio infection of Pocillopora and the impacts of temperature and light on Blackband infection of Montastria fabulata. So the mechanisms of innate immunity include having amoebocytes that are considered they're considered coral immune cells and are found in multiple sponges and invertebrates. And they act like white blood cells in that they engulf and destroy invading bacteria or other invading organisms. And also important are proteins and compounds that have direct antibacterial activity. And corals are known to produce compounds that inhibit both gram-negative and gram-positive bacterial growth. And another more recent discovery was the components of the prophenyl oxidase system, which is an important innate defense in invertebrates. And it's involved in wound healing and parasite and disease resistance, and is important in the coordination of immune responses. So also important in the innate immune system are molecular pathways that are involved in inter and intracellular signaling. This is important because cells need to be able to talk to themselves and talk to each other and understand what's happening. And if bacteria are there, are they friendly bacteria? Are they disease bacteria? There has to be a level of communication going on there. And this is all done through cell signaling molecules and receptors on cell walls. And some of these are fairly well known in other organisms and have recently been identified in corals, such as the toll-like receptor domain and complement C3-like proteins, lectins, and MAPK signaling genes. And I'm not going to go into too much detail on this diagram, but this is taken from Dunn in 2009. And it is a representation of innate immunoreceptors and protein domains in cnidarians. So when I talk about cell signaling molecules, this is what I'm referring to up here, and these would be the receptors. So this is a cell wall. And what you have is you have molecules and compounds coming in that bind to receptors and initiate a cascade throughout a pathway that provides information to the cell. So it's like downloading a program on your computer. Your computer's able to understand what's happening. So this cell is able to understand these communication signals that it's coming to it, and can then respond through the cascading pathways. Okay, so what do I mean when I say coral holobiont? Well, so you know that there's the coral host and that there's the zooxanthellae that live within the host tissue. But also important are all the little bacteria that are spread throughout the mucus layer. So, sorry, this is a very rough diagram of a coral polyp. And I put a picture of a polyp up here to kind of help you visualize it. This is a coral mouth, and this is the coral mouth with the gastrodermal cavity. And so then you see the normal coral polyps here, or tentacles, and this would be the coral tentacle. And then underneath it is the skeleton that's produced. But you find symbiotic bacteria that's important to the coral health all the way from the outside layer in the coral mucus in through the gastrodermal cavity, throughout the tissues, and down into the skeleton. And just like we have bacteria in our guts, and you may want to ignore it, but we have bacteria all over our entire body that we have to have in order to survive. We would not be healthy without that bacteria. It's the same for coral. But then we get infected by bacteria that is bad for us, or even some bacteria that's on our skin can cause an infection. The same thing happens in coral, but we just don't know the mechanisms behind it as well as we do for human disease. So what about holobiont and immunity? Well, just like we need to have healthy functional gut flora to keep us healthy, but we also have to keep our cellular tissues and our internal organs all healthy, the coral has to create some sort of balance as well. So you have the coral host, then you have the algal symbionts, and then you have the microbial community that all create a healthy holobiont. And having a balanced system creates a healthy holobiont. And when you have stressors come in, they can disrupt this balance and depending on the resistance of the holobiont, either thrust it into mortality through bleaching and or tissue loss, or into mortality through disease infection, which chews up and degrades tissues. So the hologenome theory, which is part of the holobiont theory, predicts that genetic diversity of not only microbial, but all of the symbionts involved in the coral holobiont play a role in host adaptation to stress, and that this is especially important during periods of rapid changes in the environment. So microbial diversity can aid in versatility and in the survival of the holobiont, buying it time to evolve. So as you saw in the graph of atmospheric temperature change, it's going through rapid rate of increase right now. So if through this holobiont system, corals can kind of acclimatize and just kind of ride it out, then through time they might be able to adapt and evolve to be able to be healthy in these altered conditions. And ways in which at least the microorganisms can do this within the holobiont system is through recombination and genetic mutation, gene amplification, new genes obtained from the environment, and exchanged genes from other bacteria. And this is the same thing that bacteria do with us to try to get past our immune systems. But this is hopefully going to help the coral by their healthy bacteria being able to do this and survive through changing environmental systems. So something that's going on at Harbor Branch in the Robertson Coral Reef Program currently is the study of Blackband disease infection on coral and how coral is able to deal with Blackband disease and what is the immune response, what is the mechanisms behind it. And Dr. Joshua Voss has been studying the particular bacterial communities of Blackband disease. And he's been doing this using a molecular type of technology called length heterogeneity PCR. And what this does is it reveals that there are different assemblages, so different communities within Blackband consortiums from different regions. And this is important because the bacterial communities are different, but the disease is affecting the corals the same. So it's having the same result with different communities. And that's kind of surprising. So this graph shows the assemblages parse out based on biogeographical regions. So the red is all in the Bahamas, yellow are all the bacteria consortiums that are similar to each other and from the Florida Keys, and then the bacterial assemblages from the US Virgin Islands all cluster out as well, meaning that these are all similar to each other but not similar to any of these. So the bacteria community is different. Some of the same players are there, but the composition is different, and yet it's having the same effect. It looks identical. It has the same impact on coral. So we want to know why. Is there a specific driver? What makes the infection attack corals? And even if the communities of the bacteria are different, why does it become a diseased state? And I'll actually talk about that a little bit later again in the lecture. But to get on with the final hypothesis of what's happening in coral immune systems, this is called the symbiosis suppression of coral immunity. And this hypothesis has to do with the fact that at early stages of the onset of symbiosis, zooxanthellae enter the host by phagocytosis. And in order to stay in the coral tissue, they have to block the lysozyme activity so that they're not digested. So they block the immunity of the host. This is a diagram of phagocytosis right here. So you see this would be the zooxanthellae coming in, and the coral cell would engulf it and eventually take it into its tissues. And normally the host would want to chew that up because it's not natural, it doesn't normally occur there. But the zooxanthellae suppresses this immune system so that it can stay and become part of the holobiont and become in a symbiotic relationship. In addition, genes involved in immunity are upregulated during the onset of symbiosis and then go back down. if this is true and symbiosis does suppress the immune system of coral, that's kind of surprising that it would be so dominant across all corals, and then they'd have a suppressed immune system. So it does require some further testing to understand what's going on there. Maybe it's only a temporary suppression, and then somehow there's sort of signaling that's recognized between the zooxanthellae and the host, so then the immune system does not have to be suppressed anymore. So it's an interesting theory, but it definitely requires a little bit more work, at least for me to believe it all the way. Okay, so what have I been doing with coral diseases? One of the first things I've done, I started during my graduate research and finished up here at Harbor Branch, is looking at the infection of Pocillopora damicornis with a Vibrio bacteria. And I've done this in conjunction or collaboration with Michael Morgan at Berry College and Amy Lane at Georgia Institute of Technology. So Vibrio coralliticus is a genus of marine bacteria that infects Pocillopora damicornis, which you can see here, and it results in bleaching and lysis of coral tissue. This is the coral tissue starting to bleach and starting to kind of fall off. You can see little fragments here falling off, and this is after a pretty severe onset of the infection. You can see the bare skeleton, and then the tissue is just kind of hanging on it like clothing and starting to fall off before full bleaching even occurs. And the Vibrio does not become pathogenic or severely infective until after or above 25 degrees Celsius, so it is temperature dependent. And it's thought to infect both the zooxanthellae and the coral tissue. So I did this work in Fiji on the island of Viti Levu here on the coral coast. And I collected Pocillopora damicornis by just walking out onto the reef and snorkeling and collecting fragments. And then I brought it back to the wet lab we had and infected it with cultured strains of Vibrio. Then I performed gene isolations to look for genes that were upregulated during infection, and then tested those genes during different stages of infection and at different, also under different infection conditions. And what I found was that some of the genes that were highly upregulated were, surprise, oxidative stress response genes that were they were antioxidant genes trying to deal with an intense production of ROSs in the coral tissue. And these were peroxidases and catalases that were upregulated, and the sequence homology of the genes was animal in nature, suggesting that the coral host was producing these antioxidants. In addition, another gene called carbonic anhydrase was highly upregulated, and this indicates that because of the function of the protein of carbonic anhydrase, that the cells were likely experiencing altered pH balance, and that there was issues with calcium carbonate deposition, so the corals were not able to lay down their skeleton, or they were having issues with skeleton production. In addition, there was an increased expression of potassium channel genes, and this is important because it is a gene that's involved, or a protein that's involved in initiating the apoptotic response, which is programmed cell death, as I mentioned. And it's also associated with necrosis and inflammation. And finally, an immune system response was observed in the elevated expression of sulfotransferase, which is a very important cell signaling protein that regulates host pathogen interactions. So you're seeing some sort of communication going on there regarding invaders or attackers. So what other disease research is going on here? Well, as I mentioned earlier, we are studying Blackband disease. I'm doing this in conjunction with Dr. Josh Voss, and we are doing this at in the Florida Keys, and we're artificially infecting coral fragments that have been collected from a recovered coral nursery. So not only are the coral fragments infected with Blackband, but they're also exposed to different light and temperature treatments. And then we track the disease progression to see if light or temperature is having an effect on how quickly the disease spreads across the coral colony. And Josh is measuring the bacterial community changes, and I'm going to be looking at the coral gene expression, currently doing that. And we are also looking at the coral immune activity. So this is just a diagram of some of the, or photographs of some of the coral fragments. And you can see there's different light and temperature treatments here. This is day 1 of exposure, just a little blob of Blackband disease is put on the edge of a coral colony. And then through day 4 and day 7, we track the progression and map out how far along across the coral colony, a coral fragment it has spread. And you can see by day 7, there's definitely a difference. We're seeing it, the disease encompassing at least half, if not more, of several of the fragments, whereas on this fragment, it doesn't look like it's moved really at all. So why is this? What's happening? Well, to our surprise, neither temperature nor light had any effect. There was no significant effect by either of those treatments. But what did have a significant effect or impact was which coral colony it came from. So all of these fragments were each from a different coral colony. So the colony that came from, and red, green, blue, yellow doesn't indicate the color of the colony. That's just the name we gave the colonies for identification purposes. But the fact that it came from different colonies means that different colonies had a different response means that something's going on at the colony level, and could it be immunity? So that's what we asked. We were surprised, and we said, well, why is this happening? And there's some hypotheses we have, including there's natural variability between the colonies. There could have been, some of the colonies could have had prior exposure to stress, so they already had an immune system reacted, and that immune system helped them to be more disease resistant, and that is what happens to us when we catch the flu. We're less likely to catch it again because our immune system is stimulated. Then there's also variation within and between species. So Blackband disease doesn't affect all coral species. It tends to prefer certain species. And even within those species, sometimes it's differential in its intensity. And one of the things to note is that Montastria, the species we used, has high genetic diversity within the species itself. And currently, Dr. Voss and his intern, Dan Rowan, are looking at the symbiotic community, and I am performing gene expression, and Dr. Laura Meidlars will be doing the immune response activity. So what we want to know is not just who is getting sick, but why they're getting sick, and why can some recover and others cannot. So we're trying to get a better grip of understanding the mechanisms behind resistance and resilience of coral colonies to disease infection. So what I plan on doing in the lab to further characterize this and further understand the mechanisms is I'm going to identify genes that are differentially expressed between different disease conditions, such as infected and non-infected samples, but also samples that were infected but have been able to get rid of the infection, where the infection has regressed, and they may have a wound there but they're healthy again. So were they able to get over the infection? In addition, I plan on looking at infected samples during different time points, such as between seasons, because Blackband disease is very seasonal. It's much more highly prevalent in the summer during warmer temperatures and tends to regress in the winter. And then I also want to look, as in this experiment, at colonies with different susceptibilities and try to understand what the genetic mechanisms behind the susceptibility is. And then I'll also be doing this between different diseases, such as Blackband, yellow band, yellow blotch, and Vibrio infections. So finally, the last topic to cover is ocean acidification and coral calcification. So what exactly is coral calcification? Corals grow by depositing skeleton beneath their living tissues. So this is a coral polyp, or a fragment of a coral. You can see the little different polyps there. And here's a close-up of the polyps and a diagram showing you that the gray part is their skeleton, and they extend down into their skeleton. The tissue does. So here's a more detailed diagram showing you the coral polyp and the tentacles and his gut and the surrounding tissue that overlays the coral skeleton. And then the tissue comes down in here, and skeleton is deposited here and kind of raises the coral polyp up. And then that goes on over a period of time, and as it keeps laying down skeleton and pushing itself up, it builds up, and to create one of the big colonies that we see underwater. And this skeleton is composed of aragonite, which is a fibrous crystalline form of calcium carbonate. So what exactly is ocean acidification? The changing atmospheric chemistry is what impacts ocean acidification. By having more CO2 in the atmosphere, it absorbs into the ocean. The ocean is a sink, kind of like forests are a sink for CO2 because they absorb it. That's the same thing that oceans do. Ocean environments absorb CO2, and that's natural. It's just when there's more CO2 in the environment, it throws off the chemical reaction that occurs in the ocean environment, the marine environment, to create calcium carbonate, which is what corals use to create their skeleton. So when this balance is thrown off, the ionic balance is different, and you have more protons, thus creating a lower pH. And this inhibits corals' ability to lay down their skeletons. So there's still a lot of research that needs to be done with understanding not just ocean acidification, but how it's going to impact corals, if at all. There's a lot of uncertainty there. So what people have been doing is kind of like the worst case scenario, where if it does, what are the corals going to do? And one of the positive things that have been seen is that even after severe decalcification, corals can recover. So you see here, this is a coral fragment, a coral colony fragment, little polyps, and it has a nice, pretty skeleton underneath it. And then the skeleton, through an experimental process, has been decalcified. And this is what you get. And it looks like kind of a bunch of anemones sitting on a rock, which is essentially very similar to what corals are. They are kind of like anemone that just secrete this skeleton. So then this is at a lower pH. So you have the normal pH of about 8.2, and then at a pH of 7.4, the skeleton is dissolved, and you have these polyps, but they're not bleached. They're feeding, they're photosynthesizing, so they look okay. And in fact, when pH is restored, they don't look that pretty, but they're able to put their skeleton back down and recreate it. The question is, if there was another stressor to come along, would they be more susceptible to that stress? Are they still able to reproduce? Even after they recover, are they more susceptible to stress or disease? Can they reproduce? There's a lot of questions that still need to be addressed. One of the other things we do know regarding coral calcification, or anything having to do with coral skeleton process, is there has been, they've identified the organic matrix, which is the center of calcification, and at least one protein from corals has been identified that's involved in the organic matrix. So it's very important in the production of coral skeleton, and this gene is called galaxin because it was identified from a coral galaxia. But what's interesting is, well, corals produce these big giant skeletons that can be as big as cars underwater, and we have one protein. Whereas in an eggshell, there's at least 550 proteins involved in the production of the eggshell. So there's a lot more work that needs to be done. And a few other things that we do know about coral calcification is that we know a calcium ion channel is involved because it has been characterized through molecular diagnostic processes. And we do know that there are temperature impacts to skeleton production. That's what this graph from Marshall and Claude 2004 shows. This is calcium incorporation into the skeleton, and you can see that there's an optimal temperature range. At too cold of temperatures, the skeleton process goes down, and at too warm of temperatures, it does as well. So temperature does have an effect, and that's important to know because hand in hand with ocean acidification is warming temperatures of ocean waters. In addition, we do know that coral calcification is affected by light. So this means that zooxanthellae are likely to play a role, and that's shown in this graph here. At nighttime, coral calcification decreases significantly, but it comes back on at the day, and that's an active time of calcification for the coral. So somehow the zooxanthellae are involved, and this is also important because if temperature is important and we see coral bleaching, then we'll see less calcium or less skeleton being able to be produced as well. And finally, we do know that there's a high specificity by species between the types of skeletons laid down, the ability to lay down skeleton, when they lay down skeleton, how frequently. So there is species specificity there, which means there might be species specificity in relation to recovery. So as you can see, most likely you can tell that the formation of coral skeleton is the result of complex biologically related phenomenon, and that biochemical mechanisms of coral calcification are not well understood. And on top of that, the impacts of ocean acidification are not well understood, as well as the, how intense acidification is going to be if it occurs. So we need to improve our knowledge and basic understanding of the mechanisms of coral calcification to be able to understand how they will respond to not just ocean acidification, but a lot of other stressors as well. Because as you could see in the Vibrio pacillopora example that I showed, that disease alone had an impact on coral skeleton. So future assessments of coral reef health are based on our current knowledge. And although we have made a vast step forward and our knowledge has expanded greatly in the past several decades, we still have a lot to learn. And that's where these new types of tools from molecular diagnostics can come in, become very helpful. And what we need to have a better understanding of is coral vulnerability. How well do they cope with the thresholds or limits of stress? What are their limits? How many stressors can they deal with at one time? Are there certain like lethal combination of stressors? What's their adaptability? So adaptability is slow change, and it's at the genetic level, so it occurs over a long period of time. But then there's acclimatization, which is fast, and that's a phenotypic change. And that can be a short-term solution that if they're able to acclimatize, then they're likely, if given enough time, be able to adapt to a different changing environment. And we also need to know their resilience, which is their ability to tolerate disturbance. And this is important at the colony level as well as at the ecosystem level. So finally, to understand coral physiology, we need to continue to advance our use of these molecular diagnostic tools and expand our scope of research. And like the field of molecular diagnostics with humans and model organisms, we need to move forward and take advantage of available technologies that are all around us. And here, innovation is the key. So innovation in technology use and innovation in questions being addressed is key to our understanding of where corals will be in the future. So I would like to thank the Robertson Coral Reef program here at Harbor Branch, specifically the Robertson family, who made this research and this coral health program possible, as well as Moat, Protect Our Reef, for the funding they've provided us to study Blackband disease in the Florida Keys, and NOAA and the Cooperative Institute for funding us in our future research and upcoming research to help continue to improve our understanding of coral diagnostics, and all my collaborators, including Dr. Voss, Amy Lane, and Mike Morgan. And this is one of my microarrays, and this is an unaltered image. So it's a very happy array and likes to study corals. So I will take any questions.